I'm sharing my screen. So today we will see about the before going to clinical SAS. I will explain end-to-end process in clinical SAS. So you will get a brief idea in industry how the programmers, statistical programmers develop, the analysis, statistical analysis tables. Okay, so we will see one by one. So the first step the statistical programmers or SAS programmer do is they will read the protocol document. Okay, so the first step in for a clinical SAS is protocol document. Okay, so I will show what is protocol is, the real document. Okay, and then I will show how the programmers read the protocol document. Okay. Okay, so there is a website called ct.gov. So you can see this website, clinicaltrial.gov website. Okay, so I will explain what this website is about. One minute. Okay, so I will go to this website. This website run by the National Library of Medicine. It is from USA Regulatory Authority. So whenever there is a clinical trial happened in USA, the pharma company has to disclose their study in this website. Okay. So the one of the recent topic is maybe you can find any clinical trial in this website. So there is so many options. If you want to find a clinical trial related to cancer, you can type cancer. Okay. So you can find the studies with the clinical trials. Okay. And then in left side, you will have lot of filters. So there is something like phase one, phase two, phase three clinical trials, phase four clinical trial. In phase three, you will find lot of safety and efficacy parameters. So I am choosing phase three clinical trial, and I am choosing interventional study type. And then there is something called study document. Okay, studies with the study document. So I'm choosing study protocol. That means it will filter down the studies with the study protocols. Okay. And then I'm applying a filter. Okay. So if you see here, you will find a lot of cancer clinical trials with the protocol document. So I'm going to choose any one of the clinical trial with the cancer study. Okay. I'm also choosing industry. So you will find a lot of clinical trial here. So I am going to choose the clinical trial. Okay. So here there is one clinical trial. It is for a lung cancer. Okay. And they are comparing some drug with the placebo or some other drug. Okay. So it is a lung cancer trial. Okay, and it is a phase three study. So if you scroll down, there is something called study protocol. Okay, under the more information, you will find the study protocol for the study. Okay, so protocol means it is the planning document before the clinical trial starts. the all the pharma companies have to prepare this document. So in this document, you will find when the clinical trial starts, when the clinical trial ends, okay, and when the patient has to receive the drug. Okay, so all the information will be here, why this clinical trial is conducted, and why what kind of drug the participants will receive, on what day the participants will receive the drugs. Okay, so if the programmers, SAS programmers, want to understand the study, the first step in for the SAS programmer is they will read this protocol document or planning document. Okay, so they will understand about the clinical trial study, they will understand the study design, and they will understand what is the objective of the clinical trial, and then they will start working on the programming thing. Okay, so the first step in programming is they have to read the protocol document. Okay, so for each study, there will be some programmers in pharma companies. So they will so they will work on the creating the tables and all. Okay, so this is the bladder cancer. I'm just opening some simple clinical trial which is easy to understand. Uh, Okay. So here there is prostate cancer that they are checking the safety, efficacy, pharmacokinetics behavior of luprolide mesylate. So in subjects with the prostate cancer, I think this study is looking little simple. We will open this study, okay, and then we will open the protocol document. So for this study, there will be some programmers. They will start reading the protocol document. So in the initial page, they will say what type of phase it is, whether it is phase one or phase two, and they will tell what is the study drug used. So this is the front page. And then if you scroll down, there will be something called index page. Okay. One minute. Okay, so this is the index page. You will find what are all the information available in this document. First, they will introduce about the study, why they are conducting this clinical trial, and then you will find the background information of the clinical trial, and then study rationale, why there is the need of the clinical trial, and what is the objective of this trial, and then what is the primary objective of the trial, and what is the secondary objective of the trial. Uh, Endpoints and objectives are similar. So you will find the information about the clinical trial, rationale for the clinical trial, objective of the clinical trial, study design of the clinical trial. Okay, and then what is the inclusion criteria of the trial, exclusion criteria of the trial. And the treatment plan summary means when the participants will receive the drug, whether in day one of the trial or day two of the trial. Okay. So the before the programmers start working on, they will read this protocol and then they will understand about this cancer trial. Okay. So this is just one random document which is publicly available in ct.gov. You can go to any study and open this any kind of protocol, and there you will find the information about the study. It is openly available for anyone. Okay, so any trial happened in US, you will find the trial protocol in this website, clinicaltrial.gov website. Okay. So using this protocol only, the programmers will understand about the trial. In visit one, what is happening? There is a dose administration. In visit 10, there are subjects are enrolled. In visit 13, the second dose is administered. So there will be a detailed information about this study. Okay. So I will tell what the programmers will read about. Okay. So they will go to the introduction page. Okay. They will read what is the background information of the trial. They are telling prostate cancer is one of the leading cause of the death in men globally. So, in the absence of screening, progression of asymptomatic in early stages. So, they are telling prostate is one of the leading cause of death. So, they are telling this is the reason for conducting the clinical trial. So they are telling what because of this prevalence of this cancer, they are developing a drug. Okay. So they are telling about the development of the drug leuprolide acetate. Okay, and then they will talk about some molecular level and all about the drug and the mechanism of action. You can find it here. And then what is the study rationale? Okay, so they are telling the drug was initially given by subcutaneous injection. Okay, when it was first approved in 1985. Okay, and then it is designed to deliver 22-point control over the period. Okay, and then you will find some information here. If there is any confidential information, they will mask the details in the protocol. Okay, and then they will explain why they are conducting this study. Okay, so they are telling there are 137 subjects were enrolled in this clinical trial. And then they are talking about the primary objective, secondary objective. So the programmers will read there are totally 137 subjects, and what is the primary objective of the clinical trial, and what is the secondary objective of the trial. Okay. So the programmers first they will read this planning document, and then they will understand what is the study is about. Okay. So this is the study rationale, and then they will talk about the study objective. So the primary objective of the study is to assess the efficacy and safety of the drug 25 mg for 25 weeks. So in primary objective, they are checking both efficacy and safety of the drug for 25 mg, and then they will follow up the subjects for 24 weeks. Okay, so the programmers will read this objective very carefully, and based on this only, the programmers will do the analysis. Okay, so for efficacy, they will create some tables using SAS code. For safety, also they will create the programmers will create some tables. Okay, for this data. For secondary objective also, the programmers will read the data. The secondary objective is about the pharmacokinetics of the drug. So the primary objective of the trial is to assess the efficacy and safety of the 25 mg of the drug. And the secondary objective is about the pharmacokinetics. So the programmers will develop the tables, or they will do the analysis for efficacy, safety, and pharmacokinetics. So the programmers will read about the objective carefully before starting any work. This is the planning document. Okay, then they will tell in under efficacy what are all the table they will generate. They will generate mean for prostate cancer, right? They will check the testosterone and LH levels. So they will calculate the average changes in testosterone and LH levels. Okay, and then they will also check the effect of the drug 25 mg from change of the antigen levels. Also they will check, and also they will check serum LH levels. So this and all they will create a separate table for the average and the change in antigen level and change in serum LH level. For each, the SAS programmers will develop a table for this. Okay, so this is and all before the trial starts. This all this planning information programmers will read carefully. Okay, then they will understand the design. Okay, so they will understand the study is primarily about the safety, efficacy, and pharmacokinetics of the drug. And for under safety, you will they are given they will have a table for adverse event, serious adverse event, okay, and change in bone, okay, and pharmacokinetics and all. And in pharmacokinetics, what they will analyze Cmax, Tmax, maximum concentration, and the maximum time period. Okay, area under curve. Okay, so all this the programmers will read, and exploratory is additional analysis. Okay, and then the here the comes the very important thing, study design. Okay, so here the study they are saying this is multinational. The study is happening in a multiple country, multiple center. Open label means there is only one drug, so the participants will know what drug they are receiving, and since it is a prostate cancer, all the subjects will be male with the cancer. Okay, and then they are talking about. Some inclusion exclusion criteria. So the subjects whose testosterone level greater than 150. So they are talking about some inclusion exclusion criteria, and then they are saying the study happens for 24 weeks. That is roughly 5 6 months. Okay, in the 6 months, the two doses of the drug will be given to subjects by the subcutaneous injection. So the first one is 12 weeks, and in another 12 weeks, the second dose will be given. Okay, the first dose is on day zero, and the second dose is given on the day 84. Okay, at the end of 12 weeks, all the subjects will be administered second dose of mg, and then they will be followed. Okay, so the programmers will read this carefully, and so they are telling on what day and at what at what day the participants will receive the drugs. Okay, so this and all happens before the clinical trial starts. Okay. So the in protocol, all the planning information will be there, and there will be inclusion criteria for the trial. So only the participants greater than 18-year-old will be enrolled or will be eligible to participate. Males with any histologically confirmed, only the cancer patient will be enrolled. Okay, and the testosterone level should be greater than 150. Okay, so there will be some inclusion criteria. Similarly, there will be some exclusion criteria. Anything that comes under this, they will be omitted from the clinical trial. Okay, so there will be inclusion exclusion criteria given. Okay, so and then this is the treatment plan summary. So treatment plan summary, they will tell on what is the drug, what is the composition, and what day they will receive, and in second dose what they will receive. So in treatment plan also you will understand about the schedule of the drug. Okay, and what is the rationale for the dose. So the programmers will read this very carefully, and then they will understand the study designs. Okay. After that, so here they will have the study schedule. The first dose will be given on first visit. No, the first visit is on screening. They will do all the inclusion criteria, exclusion criteria. They will enroll the subject, and in visit two, the first dose will be given. Okay, in visit three, visit four, they will take the blood samples. In visit three, they will take the blood samples and they will check the safety parameters, whether they have any adverse event or not. They will check the sugar level. So in each visit, they will take the blood samples, and then they will check the antigen level, they will check the sugar level, they will check the blood pressure level. So they will take various blood samples at each visit, and then they will check the safety efficacy parameter. Even after second dose will be given at the 13th visit, that is day 84. Okay. So here you can see what is the day, what is the week they are getting the dose and all. Okay. So here you will see what are all the examination will be taken. So here, so at each visit they will take the ECG, vital signs. So in the day, okay. So in day one also they will take some vital signs. So you can find when they will take ECG, when they will take vital signs, when they will give concomitant treatment, when they will collect adverse events, and all this information will be given under this one. Okay, so the programmers has to understand this study schedule also. So you can also download and find this information. Okay, 1 minute. Okay. So based on this only, the programmers will understand the study design. So whenever the data is collected for your, if you see the in day three, the vital signs are collected. Okay, in day one, vital signs are collected. Day two, vital signs are collected. Day three, vital signs are collected. Similarly, the ECG is taken at day screening, and ECG was taken at yeah, fourth hour, and the ECG is taken at day 28 also. So whenever they take ECG, they will take the data also using CRF. So whenever they take some blood samples or when they collect some vital signs, the data will be entered in the database. Okay. So these are all the data collection from the patients, vital signs, ECG, in okay, an adverse event will be collected in day wise. All the adverse event data will be collected day one, day two, day three. All the day the adverse events will be collected. Okay, so the programmers will understand about the study schedule very nicely from here. Okay. So the from this the ones who read the protocol, they will understand the clinical trial in a better way, when the trial starts, when the trial ends, what kind of data will be taken, what is the objective of the trial, what is the primary objective of the trial, what is the secondary objective of the trial. Okay. See, apart from that, they are taken urine analysis at screening. They are taking urine analysis at day one, and they are taking urine analysis at day two also. Okay. So any trial, you can find the protocol here. Okay. So here they will explain at a screening visit, day one, what they will do. They will collect the informed consent, and then they will collect the data about inclusion exclusion criteria. So what kind of data they will collect? For inclusion criteria, they will collect the testosterone levels. They will take hematology test. They will take some biochemistry test. So at each visit, they will tell what are all the data has to be collected. Okay, at visit two, day one, at the time of administration, what the subjects has to do, what has to be done, this information will be your, so each subject, the enrollment number will be given, and then they will vital signs will be collected, ECOG, and then they will do the ECG, and then at day one also they will take the hematology biochemistry. So before dose is given, they will take the blood samples and they will check all the parameters, safety, efficacy. After drug is given, they will also check the biochemistry, hematology, or for safety, efficacy. So even before drug is given, they will check all the parameters, safety, efficacy. After given the drug also, they will check all these parameters. At each time, the data will be collected. Okay, what is their antigen level, what is the blood sugar level, whether any adverse event is recorded. So at each visit, the all the parameters will be collected in the database. Okay. So all the information you will find visit one, visit two, what data will be collected. Okay. So this is so this also the programmers will understand about the date. For understanding the data, they will read this information. Okay, so study assessment, it is fine. You need not to read. Okay, there is one information called statistical analysis section. Okay, 1 minute. Mm. Okay, so there is section called 10. So it will talk about the statistical consideration and analytical plan. Okay, so this is the important section for statistical programmer or SAS programmer. So they will tell how many, how the I have initially told there are 133 subjects are expected to be enrolled, right? So the before the clinical trial starts itself, they will tell how many subjects has to be enrolled. Okay, based on the sample size only, they will enroll the number of participants. Okay, so before the trial starts itself, the statistician or statistical programmer will come up with this number 133 subjects. They are expected to be enrolled in the study. Okay, and they will talk some statistical terms. Okay, here. So if you want to know what is the sample size of the clinical trial, you can find it in this statistical consideration section. And what is the analysis population? Okay, analysis population means I have told there will be some primary objective, secondary objective, right? The primary objective of the trial is safety and efficacy. So, okay, for assessing the, for analyzing the safety or efficacy, what kind of population will be considered? Okay, so here there are two kind of population. One is intent to treat. Intent to treat means the population consists of any subject receiving at least one dose of drug. So we will analyze the data for the participants who receive at least one dose of the drug. Okay, if the participants data has to be analyzed, they should have at least received one dose of the drug. Okay, there are another population, per protocol population. This population means they would have received two doses of the drug. Okay, intention to treat means they would have received at least one dose of the drug. Per protocol means they would have received two doses of the drug. Okay, so for some for some primary objective, they will do the analysis only on the intent to treat population. For some secondary endpoint, for some primary endpoint, they would have done the analysis on the per protocol analysis. Okay, So here they will tell what is the analysis method to be used. So here they are telling they will analyze mean, standard deviation, median, okay, and categorical variable. They will do the t-test Wilcoxon sign. Okay, so based on this only, the programmers they will have the data, and based on this analysis method, the programmers will write the code. Okay. So, under efficacy, you will see what is the primary objective, how the primary objective has to be analyzed. So, they are telling the percentage of subjects with the testosterone concentration. For this objective, how the data should be analyzed. They are telling it will be analyzed using the binomial distribution method. Okay, so they will tell how the primary objective has to be analyzed statistically, how the secondary objective has to be analyzed. Okay, so they will tell what is the statistical method it has to be used. For that, the programmers will write the code. Okay, so before doing analysis, the first thing the programmers or anyone in the clinical trial do is they will read and understand about the clinical trial. Okay. So for safety analysis, they will tell how to analyze the safety analysis data. Okay. So the first step the programmers do is they will read the protocol. The pharma company will give the protocol to the medical writing team uh, the statistician programmers to everyone. Okay, so, and after some time, the in ct.gov website, the pharma company has to disclose the upload the protocol. Okay, maybe 1 year after the study ends, the pharma company has to disclose the study protocol. Okay, as per the regulatory authority, USA government. After the trial completes within 1 year, the pharma company has to disclose this study protocol. Okay. So the main protocol, the purpose of the protocol is to understand what is the primary objective, what is the secondary objective, what is the study design, what is the schedule of visit, what is the treatment, inclusion, exclusion criteria, sample size, if there is any safety monitoring. So the main thing is the first step in the programming is they will understand the protocol document. Okay. Okay, so this is the same. They have given Adam and all. We will come later stage. Okay. Similarly, there is one more document called statistical analysis plan. Statistical analysis plan is also a planning document. Before the trial starts, the statistician developed this. Study protocol talks about a lot of general things like study design and study rationale, but it will talk about the statistics in very limited way. But if you see statistical analysis plan, it will talk about the statistical thing in a very a huge way. Okay, so the majority of the document contains the information about the statistical methods. Okay, so whether any general statistical issue, any how to handle the data, if there is any missing data, how to handle that. Okay, what kind of population has to be used for the objective. So the majority of the documents talk about the statistical analysis. In protocol, it is a general planning document. There also they talk about the statistical method, but in a very limited way. So before the trial starts, the programmers read both the protocol and statistical analysis plan. Okay, so this is the first step. Okay, before the trial starts itself, the pharma company developed this planning document. Okay, so before that, okay, so this is the first step. Okay, anyone in the clinical trial, they have to read the protocol and analysis plan. Then only they will understand about the clinical trial in a better way. Okay, so this is the first step. After the second step is okay. So we have seen one sample size, right? Okay, let me show you once again. We have seen sample size, right? Okay, sorry. So there is something called sample size. Okay, so the sample size, if they in planning document, if they say 133 subjects, they will enroll only 133 subjects for the clinical trial. Okay, so there will be something called inclusion exclusion criteria. 200, 500 subjects will come. Only if they align with this inclusion exclusion criteria, they will be enrolled in the trial. Okay. So once 133 participants enrolled in the clinical trial, the day one, day two starts. Okay. The screening visit will start. And then in day one, they will collect the blood samples. Day two, they will give the dose one at the day one. Okay, so only when the participants align with this inclusion exclusion criteria, they will enter in the clinical trial. The data will be collected from them. Okay, safety, efficacy, and pharmacokinetics parameters. Okay. Okay. So now this is the first step. After that, there is something called okay. There is something called CRF. Okay. Uh, Okay, so there is something called CRF. Okay, what is that means? It is the questionnaire, it is the form where the data is collected from. Okay, for each subject, okay, they will give this form to the subjects. Okay, and then they will ask the questionnaires. Okay, for example, uh, for each inclusion criteria, they will have each question whether the subject is male or female. If it is male, then so from initially the day one or day zero, 500 subjects will come. Okay, for each subject, they will collect this data using this questionnaire. They will ask whether he is male or female based on the inclusion criteria. They will have this questionnaire whether they smoke, whether this is another questionnaire. Okay, for our sub study, the questionnaire will be different. Only if they satisfy with the answer, the subjects will be enrolled in the clinical trial. Okay. So for each subject, they will ask this questionnaire at the screening time. Only if they align with this criteria, they will enter in this clinical trial. Okay. So this questions will be asked at the time of screening visit. Okay, that is day zero. Okay. Once they enter in clinical trial, there will be something called CRF. Okay. In CRF also there will be lot of questionnaires. Okay, I will show that what kind of questions they will ask. Okay, so we will see one CRF. So this is for the pancreatic cancer. Okay, so if they want to collect about the medical history, they will ask this questionnaire. Okay, so they will ask whether the okay, what is the date of pathologic diagnosis? Does the subject have the pancreatic exocrine insufficiency? So they will have some electronic questionnaire. Okay, so this is the electronic questionnaire. So they will collect so many parameters like the safety. They will have set of questions for efficacy. They will have set of questions for pharmacokinetics. They will have set of these questions. Okay, so for each domain, for safety, there will be 50 set of questions. For efficacy, they will have 50 sets of questions, and each data will be entered here subject wise, and then it will be entered in the database. Okay, in day one, the once the dose is given, the data will be collected. ECG, for ECG also there will be some questionnaires. Urine analysis, there will be some questionnaire. So for each thing they will have some questionnaire and the data will be entered. Once the data is entered, it will go and sit in the database. Okay, so CRF in clinical trial, there is something called CRF. Okay, CRF means it is the form. Okay, case report form. So in case report form, they will enter the data. Okay, data about demographic data, efficacy data, safety data. Okay, in screening visit, they will collect the data about inclusion exclusion criteria. Once the subject is enrolled, they will collect the data about safety, efficacy, and pharmacokinetics. Okay, so at each visit, they will collect the data using CRF. Okay, in screening, they will check the data about inclusion, exclusion. In day one, the blood samples, efficacy, safety, so many parameters, it will be collected for subject wise. Okay. So, as I mentioned, this is the questionnaire case report form. They will ask questions about the study visit, okay, and then the data will be collected for day one, day two, day three for each time. Okay, so this is basically a questionnaire, electronical questionnaire. The data will be collected for each visit or each time points, day one, day two, day three. Okay, so this and all some advanced concepts we will cover it in a later stage, STDM, ADAM, and all. Okay. Okay, so the first one is the programmers read the protocol and then SAP to understand the trial, and then or anyone in the clinical trial before the trial start, they will read this planning document, or the person who is in the clinical trial site, doctors or clinical research assistant, they will also read this document. Based on this document only, they will conduct the clinical trial. After that, using questionnaires, they will collect the data for safety, efficacy, demographic, inclusion, exclusion criteria, and then the data will be stored in the database. Okay. Once the data is stored in the database, the programmers will develop a data for demographic. The programmers will develop a data tables for adverse event. The SAS programmers will develop a table for efficacy tables, pharmacokinetics tables. So we have seen primary objective, secondary objective, right? So based on that analysis, the SAS programmer will develop safety tables, efficacy tables, demographic tables. Okay, so this is a chart of this is general workflow. First protocol SAP, and then the pharma company collect the data using case report form. After that, they will develop different types of tables, safety, efficacy, and pharmacokinetics tables. Okay, so this and all some specialized technical terms for those tables. Okay, STDM, ADAM, TLF. Basically, it are all the SAS tables. Okay, so in futures, we will start with the protocol app, and then we will see how the data is collected. and then how SAS programmers created the safety, efficacy, and pharmacokinetics. So we will see each tables, okay, how pharmacokinetics tables are generated, how safety tables are generated, how efficacy tables are generated. Okay. Okay, so that's all from my side. If you have any questions, let me know. Otherwise, we will see in next class. Uh, I have not given the assignment, the project for basic. Maybe at the end of advanced SAS, I will give the combined project. But for assignment, I will give one assignment for this session, how to read the protocol. So basically, you need to go to ct.gov and then you have to open any study, and then maybe I will give you assignment to give a summary about the clinical trial. You can open any HIV trial, you can open any infection or any kind of clinical trial you open, and then you have to select some protocol, and then you have to summarize about the study design. Okay, maybe I will give some small assignment for this by today or tomorrow. Okay. So from next week onwards, we will see about how programmers develop the tables. Okay, using code, SAS code. Okay, okay, that's it from my side. If you have any questions, let me know. Otherwise, we can close the session.
So in the clinical trial dot government, if they give us the NCT numbers, can we directly search of the related paper?
Yeah, yeah. So each trial it has NCT ID. Using NCT ID, if you know the NCT ID, you can go to the okay. So if you know the NCT ID in front page of ct.gov, in there is something called other terms, so you can search using nct id. So here also you can search using nct id, and if you click search, that particular study will come. Okay, so from there also you can select the particular study and download the data. But generally what people do is they will in other terms they will search hiv, covid or something, and then they will select the particular clinic. You can search by NCT ID also, whatever you're convenient. Yeah.
Okay, sir.
Okay.
Sir, can we calculate the sample
Oh,
size using SAS?
yes, yes, yes. Definitely the in all the pharma companies they use the sample size calculation using SAS only. So have you heard about Z test, T test, and all, right? So from Z test, T test only, from that formula they will have something called effect size. Okay. So when they are comparing the treatment with placebo, they will know what is the effect size, what is the efficacy between treatment and placebo, whether the treatment is superior to placebo or placebo is superior to treatment. So they know that treat effect size. Based on that effect size, they will calculate the sample size. So there is some procedure in SAS called PROC is a test, PROC T test. From that procedure, they will calculate the sample size. In our class, we will see about that sample size calculation. In 2 or 3 weeks, we will see about that. Yeah. Okay. Oh.
Will you go in detail about the city standards?
Can you repeat again your voice?
Will you go in detail about city standards?
This website, do you mean?
No, no, cities.
C disk. Okay. Yeah. Okay. So you want to know about this website or what is your
Yes,
question?
yes, yes, yes.
So C disk is nothing but so okay, I will explain with one simple example. Uh, okay. Okay. Let me close this. So I will open one Excel. I will tell what is the need of C disk. So C disk is nothing but okay. So as I said, in pharma company, they use the CRF to collect the data. Okay, maybe one domain is they will collect the data about demographic. Okay, uh, for example, for each subject, they will calculate the data about age. Okay, so here they will collect the data about age. Here they will collect the data about gender. Here they will collect the data about race, ethnicity, and all. Okay, what C dash tell us, whichever pharma company they are, irrespective of pharma company, they have to use the standardized variable name. Okay, gender means they should not use sex. Okay, the variable name should be standardized, and what are all the variable names should be in each domain. If it is demographic, age, gender, race, ethnicity should be definitely there, mandatorily there. If it is a safety, serious adverse event, non-serious adverse event, what are all the variables should be here. It tells about what is the variable names to be collected, and what is the variable name to be used in the domain, and what is the character limit about the variable. Okay, for each, what is the character limit. So it is the standard for collecting the data. Okay, cdisk is nothing but it is the standard used in the collecting the data and also for developing the tables. Okay, so it is simply in raw data, this variables and all is not in the standardized form. Okay, what programmers will do, if there is sex, they will convert this variable name into gender. Okay, in the raw data. So it is just a standard for developing your tables, clinical trial tables. Got it?
Mm. Yes, yes.
Okay,
Yeah.
okay.
Is how this CD standard connect with SAS?
When SAS programmers, so in raw data, what the programmers will do is the CDIS standards will have guidance. Okay, for each
Oh, okay, got it.
domains, they will have some guidance. For
Guidance.
demographic, they will tell five variables to be used, and the character limit has to be five, six, seven. So based on that, the programmers will write the code to rename the variable, or they will merge some variables. So they will update the variable date column. So
Mm.
they will the based on this guidance only, they will work on the programming part.
Okay, got it, got it. Okay.
So they
Thank
have a nearly for 40 different domains, they have a guidance, okay, in CDISC for demographic, safety, efficacy, vital signs, ECG. For each parameter, they have a guidance, CDISC.
Mm, okay.
Okay,
Thank you.
okay, okay. If there is no other questions, we will connect next week. Thank you, everyone. We will see next week.
Thank you. See you.
Thank you. Bye.